Hello again. This is part one of the immune system. Now, the immune system is very complex, so covering it in two videos is just sort of skimming the surface. If you really want to learn more about the immune system, probably an entire semester class would do. But I think this will provide a very good introduction to that. Cells and molecules of the immune system are circulating throughout your body all the time, but then when you're exposed to a respiratory disease such as coronavirus, the SARS-CoV-2 or other coronaviruses, and influenza, your immune system starts to recognize those infections because of the proteins that are on those organisms. That are different from the proteins on your cells and in your cells. You can also be exposed by cutting through the skin. Wounds, cuts, punctures, things like that will introduce pathogenic organisms usually. Eating things that have pathogens in them, transplants such as blood transfusions and other types of transplants. And then you have allergens, which will affect the immune system in different ways. So they sort of overreact in some cases to particular epitopes of proteins, that is, the surface sections of some of those proteins. So when you are exposed to these antigens, as I said, they're mainly epitopes of proteins or portions of proteins. You have a first exposure, and there's a lag time for T cells and B cells to become active. And there's actually something happening here at the beginning that I'll talk about later in this lecture, and that's innate immunity versus acquired immunity of the T cells and B cells. So there's initially this high reaction of T cells, and then those eventually go down as the infection goes down, as these T cells and the B cells start to kill that organism and clear the body of those antigens. Then you can have weeks later an inapparent reinfection and see those T cells and B cells rise again, but they've already worked out what that epitope is, so it takes them a shorter time to deal with that infection. And then mild reinfection or inapparent reinfection due to memory cells, T cells and B cells that have been saved in the lymphatic system, and so you can become immune for years and years for many of the antigens and pathogens. Whether this graph applies to the SARS-CoV-2 virus, no one knows quite yet. The main organs and tissues of the immune system, of course, many of the blood cells are formed in the bone marrow, and not only in the long bones such as the femur, but also in the hips. So there are a couple of different locations for that. Then you have areas of the lungs and the intestines, where they have little patches on the epithelial layers, which interact with the antigens. The thymus also is a place where the immune system is essentially trained not to kill your own cells. Spleen is also an area of activity, and then the lymphatic system, tonsils, adenoids, and lymph nodes, which is where a lot of the memory cells are taught to react to certain antigens, and there are cells that are called memory cells that are stored there in case of an additional infection later by that same antigen. Here's another diagram, and I just put this up mainly to show that the lymphatic system goes further than what was shown in the previous diagram, so that that lymphatic system goes throughout the body, and that is a way to move these B and T cells around in, as well as other blood cells around in the body for the immune response. So, as I said, there's sort of a lag period between when you're infected and when the T and B cells really work out what the antigen is. And the early sections are handled by this innate immunity, which controls inflammation, fever, and opening up essentially the blood vessels to the infected area, swelling and things like that. Adaptive immunity has to do with those T and B cells, and so you have two types of immunity, one that kicks in very soon and is also responsible for a lot of the allergic responses, and then adaptive immunity, and there's both natural and artificial. The natural is gaining that immunity maternally by the fetus and the infant who's drinking the mother's milk, and then active by an infection. And you also have artificial immunity, adaptive immunity, one through antibody transfer, through blood transfusions, etc., and then active by immunizations, vaccinations. And it's really these two active ones where you get long-term immunity. The other ones, the passive ones, there's only transient immunity usually. So innate immunity here, indicated by this sort of purple circle, and adaptive immunity work together, and in some cases cells overlap between the two. So there's sort of an initial response, rapid by innate immunity, accomplished by several types of white blood cells, including dendritic cells, mast cells, macrophages, natural killer cells, basophils, eosinophils, neutrophils, granulocytes, and the complement system, as well as some T cells. And then adaptive immunity is really exclusive to T cells and B cells, and T cells come in many types. The B cells are the ones that form a lot of the antibodies, or most of the antibodies, and they can appear on the surface of the B cells, but they can also secrete those into the lymphatic system, into the blood system, to clean up, essentially, or attach to those antigens. There are many types of immune cells, as I indicated previously, and depending on what type of organism infects, whether it's a virus or a bacterium or a parasite of some sort, the immune system will react differently to those by stimulating the growth and differentiation of certain cells and inhibiting others. So here you have an immature dendritic cells. Now, dendritic cells present the antigen over here to the T cells, and these are Th, or T helper cells. These are central to the immune system in then sending signals to the rest of the immune system on what to do, what type of organism is infecting. So if it's bacteria or viruses only, this will signal a certain type of T cell to become this type of T cell, Th1, and it'll produce certain cytokines, interferons and interleukins and things like that, which are molecules that will then kill or interact with T and B cells and other types of cells to stimulate or repress different parts of the immune system. On the other hand, when you have infection by any of these, including parasites, then it'll signal a different type of T cell here, a naive cell, which will stimulate the production of regulatory T cells, which will also produce interleukins and specific growth factors. And it will also inhibit the differentiation and growth of a couple of other types of T cells, since they're not needed in this reaction. If it's only parasites, then it'll signal this pathway with a different type of T cell that then will stimulate Th2, the T helper cells type 2 in the immune system. And those form different interleukins, which will then signal other parts of the immune system, both T cells and B cells, as well as other types of cells of the immune system to react in a certain way, proliferate, not proliferate, kill certain types of antigens, etc. So this illustrates one of those complex interactions when a parasite infects a human or other organism. Immediately there's a rapid expansion in the number of eosinophils, mast cells, and basophils, and those secrete interleukins, and T helper cells also secrete interleukins. Those cause recruitment of mast cells, and the mast cells here produce histamines, and they also are responsible for inflammation, increase in blood flow in the area to sort of release other white cells to attack the foreign antigens. There are also cells down here, other T cells, that also produce interleukins, and those are to suppress certain T cells and other parts of the immune system, including mast cells, so that inflammation doesn't get too extreme. Also, they're responsible for mucus production, and this is one thing that in hyperimmune responses, you get fluid retention, including fluids in the lungs, such as what's happening in the COVID infections or SARS-CoV-2 infections and influenza too in extreme cases. So the fluids, then the lung fills up with the fluids and the people essentially suffocate. So in normal circumstances, the mucus is there to spread some of the antimicrobial and other kinds of compounds, as well as cells, to coat the system essentially with immunological molecules and immunological cells. But when it's hyperactive, then it causes some problems in creating too much fluid. So one of the first things that happens in an infection is you have an innate response here. Those respond to antigens, bacterial or otherwise, at the surfaces. So on the surface of the skin, in cuts, in the lungs, in the intestines, and elsewhere in the body. So those interact immediately to try to bring the blood supply to that site to release more of the immune cells and cytokines and things like that to stimulate the immune system. And then there's the adaptive response, where you have dendritic cells. These are cells with many, many surfaces, and they present then those antigens on the ends of these extensions of the cells. And they present those to T cells of two different types, CD8 and CD4, as well as other T cells. The CD8 cells then produce cytokines, interact with macrophages and natural killer cells, and go back and attack the bacteria or whatever organism happens to be infecting. Whereas the CD4 T cells will then also stimulate macrophages and natural killer cells, but they'll also stimulate the B cells, and to start them on a pathway to find the correct antibody that will attack or attach to that antigen on the invading organism or antigen. And then some of those will further differentiate into plasma cells, which will release those antibodies in the bloodstream and the lymphatic system to attach to more of those antigens and invading organisms. In the lymph nodes, some of each of these types, the CD8 and CD4, type cells will be signaled to become memory cells in the lymph. And also some of the B cells will become memory cells, be induced to stay in the lymphatic system. And then if that antigen ever enters the body again, then there's a fast reaction of these rather than trying to work out by recombination and expression of those genes, trying to work out what type of antigen that is. These ones already know what the antigen is, and they can immediately go into action to attack it, and neutralize it, and kill the organisms that are invading. So here are a few more details about innate immunity. So if a bacterium or a virus or some other organism invades a cell, then the complement system can immediately attack those organisms and lyse them, or kill the cell that contains them. These also will signal other things such as macrophages, mast cells, and leukocytes, neutrophils, eosinophils, basophils, and granulocytes, which will increase inflammation. It does two things. It brings the blood supply there, and it also heats the area. So a lot of these organisms won't be able to divide in there because they're heat sensitive. These also will attach to other cells and activate other types of cells. Dendritic cells will attach the antigen to themselves and then present that to T cells. The T cells then will activate certain B cells that match that antigen. They can also phagocytize either the bacteria, the viruses, or organisms that contain those organisms, bacteria and viruses, and kill them. This can be done by natural killer cells also, which move up beside and attach to a cell that's infected and actually just lyse it. Some of the videos of those are kind of interesting, where the cell just dissolves before your eyes. And then you go into adaptive immunity here, where the T cells then will signal other types of cells, including B cells and other types of T cells, to concentrate on a certain antibody type, which will kill whatever invading organism has that particular epitope of the antigen or the protein, that is the part of the protein that is exposed on those organisms. So that complement system, which is involved in innate immunity, one of the first things to occur, can proceed by several pathways. One is the lectin pathway, so it attaches certain proteins to that and will cause other parts of the complement pathways to activate. Then there's the classical pathway that goes through several steps to eventually cause signaling of a host cell, such as a T cell or a dendritic cell, to become active, indicating there's an infection. It can also go through an alternative pathway here that causes cell lysis of the pathogen cell, poking holes in the cell membrane. However, there are some pathogens that have developed some ways to inhibit this pathway, either by capturing certain molecules or by cleaving certain molecules or by inhibiting particular pathways. So in some cases, the pathogens can evade the complement system, at least partially. So that C3 protein is especially central to the complement system. It's right in the middle here. So you have the classical pathway leading into C3 and eventually lysis, inflammation, or opsonization, that is, marking of those cells for capture and destruction. And then there's a lectin pathway that attaches mainly to carbohydrate groups on the cell membrane, and then an alternative pathway. But they all lead into this C3, which is central. So those organisms that can destroy C3 can avoid parts of the complement system pathways. Some strains of Staphylococcus are able to do that by destroying C3 or sometimes C5 and other complement system proteins. Innate immunity responses are performed primarily by myeloid cells, or things that come from the common myeloid progenitor here. And then you have a erythroid progenitor. Some of those become erythrocytes, some become platelets, and then other types of cells also come from those original cells, including basophils, neutrophils, eosinophils, monocytes, and monocytes then mature into macrophages and dendritic cells. And these all have functions in innate immunity by presenting the antigen to other cells, including the dendritic cells do most of that or much of that. Macrophages that run around and signal other cells, and also phagocytize bacteria, viruses, and infected cells. Basophils are responsible for inflammation, so they dilate blood vessels, and they also cause extension of blood vessels, which does two things. It increases the blood supply in the region, and it also heats up the wound or wherever the organisms are coming into the host, so that you get a localized heating of that area. They also produce histamines and serotonin and heparin, which reduces blood clotting. So they bring a lot of blood in that area, but they don't allow clotting, so that you get a lot of blood cells recruited to the area. And one of the other white blood cell types, neutrophils, are attracted to places of inflammation, and they're in the highest concentrations, up 50% and sometimes more of the white blood cells are neutrophils. They are the major thing that you find in pus, because they phagocytize the invading organisms and the antigens of those organisms. So in pus, then you would find mainly neutrophils that are filled with the pathogenic organisms. Eosinophils are in very low concentrations, and they react primarily to parasites, although some also will attack viruses as well. These ones produce reactive oxygen species, that is, things that when they're released from these eosinophils will essentially oxidize and kill the invading organisms, most of which are usually anaerobic. And all of these can also release certain types of molecules, such as cytokines and interleukins and things like that, which signal other parts of the immune system to the site and to become active as well. Macrophages also phagocytize bacteria as well as different antigen particles. Dendritic cells actually take those organisms into the cell and then break the proteins into little pieces, and then present those little pieces at their cell surface and present those to other cells, such as macrophages and T cells. On the other side here, you have the lymphoid cells, including natural killer cells, large granular lymphocytes that sort of act in between the innate and acquired immunity systems. And then there are these small lymphocytes that mature into T lymphocytes, B lymphocytes, and the B lymphocytes can further differentiate into plasma cells that then release the antibodies that they're forming into the fluids of the body, including interstitial fluids, blood, lymph, mucus, and others. And those are formed primarily in the bone marrow, as are all blood cells. But they wind up in two different lineages. These are T cells, which is where the T comes from, and then the B cells from the bone marrow. The B cells, of course, become mature lymphocytes with specific antibodies sticking out of their cell membranes. And then some of those can mature further into plasma cells, which release those antibodies into the fluids. Some of these then circulate through the lymph nodes, through the spleen, and other locations. They can also circulate around through the blood, so they can enter those different compartments in the body and then recirculate through the blood, and then thus sort of covering all bases as far as where they're circulating to react with the foreign antigens. And this is true of both T cells and B cells. So as I said, the innate immune system kicks in first, and you get this rapid increase in interferons, both interferon alpha and beta types. And this will inhibit mainly virus infections, but also some other types of organisms. You also get an increase in natural killer cells, which essentially move up next to one of the infected host cells and will essentially kill it. So that's why sometimes you feel a little ill, because some of your cells are being killed and they're releasing toxins into the surrounding fluids. Then the virus titer in this case is starting to decline, because you're actually killing it in here with the interferons, which are signaling parts of the immune system, the white cells as well as the complement system. And then after a few days of infection, you get an increase of these cytotoxic lymphocytes, including T and B cells, and an increase in the amount of antibody that's specific for that virus. And so that eventually resolves the infection, or at least you hope that resolves the infection. So here's another diagram to look at that. If you have an attack by the pathogen, some macrophages can chase bacteria and things like that around and attach to them, and then they take up the pathogen and chew it up, essentially dissolve it. They also, parts of the pathogen can attach to a dendritic cell and be presented to a T cell down here, but also when they attach those pathogenic or foreign antigens, they can start an inflammatory cytokine reaction. And that can cause inflammatory reactions, including shock, this is one thing that's happening with the SARS-CoV-2, is that you're getting essentially a cytokine storm. It's a hyperactivity in this, which causes shock in some patients. They can also release interferons, which also attack viruses, but also are responsible for some autoimmune diseases. When they signal a T cell, they can do it by either attaching to or producing specific cytokines, or both. And in that case, you can get differentiation of these T cells into type 1 and type 2. These can signal cytotoxic cells to attack those particular organisms, viruses, bacteria, or tumors in some cases. And the Th2 cells can signal B cells to attack by antibody release, but they can also cause allergic reactions as well. So next I want to talk a little bit about the lymphatic system, which is where many of these different reactions between cells, signaling between cells, occurs. While the white and red blood cells circulate in the blood, once they've reacted with an antigen or somehow been activated by interaction with an antigen or another cell of the immune system. Then they enter the lymphatic system and drain into the lymph nodes. Here it is shown diagrammatically when there's an infection, in this case by a bacterium, but it also could be a virus or a eukaryotic cell also. Then there's a reaction by these dendritic cells as well as other lymphocytes that react with the proteins on those invading organisms and essentially digest some of the proteins. Some of those protein parts go directly into the lymphatic system, and some of the dendritic cells also go into the lymphatic system, carrying, displaying essentially, or presenting those parts of those proteins, the epitopes of the proteins. And again, epitope just means surface portions of those proteins. So here's how each one of those progress. The T cells that enter the thymus are then trained not to kill any of the proteins in the host organism. And then there's a naive pool of these CD4 and CD8 and other T cells. And then eventually in the lymphatic system, then these dendritic cells, then or antigen-presenting cells, APCs, so they can be other than dendritic cells also, signal the CD8 and CD4 cells to react whenever they see that protein again. So there's a short-term immunity and a long-term immunity that develops in the lymphatic system. And there are similar reactions with B cells, which also enter the lymphatic system and then react, are stimulated by the antigen-presenting cells to become memory cells, and they also can differentiate into plasma cells that instead of just displaying the immunoglobulin on their membranes, on their surfaces, they release those immunoglobulins into the bloodstream, as well as other areas of the organism. So this shows a little more about that process, where then the dendritic cells here, which have a lot of membrane surface and have cell surface proteins that present the antigens, but they recognize a foreign invader. They take that invader, they phagocytize it into the cell, and then start breaking off pieces of the protein and presenting that protein out on these receptors. They then migrate into the lymphatic system, so they go in between cells and make their way into the lymphatic system. And then once they reach a lymph node, they start activating and communicating with T cells. But they also interact, as you can see up here, with macrophages and with B cells. And the macrophages primarily take up some of those broken pieces of proteins and present those at their cell surface, and those also can communicate from one type of immune cell to another type of immune cell. And this shows some of those interactions. So here you have this dendritic cell that is presenting the antigen to its cell surface and presenting that to a naive CD4 T cell. That activates this CD4 T cell to also activate the appropriate B cells that have that same antigen on their surface. So then you get proliferation of those B cells with the correct antibody on their surface that will react with that antigen that's just been presented by the dendritic cell and by the B cell here. Some of those are stimulated to then develop into the plasma cells, which will then produce and release a lot of that type of antibody, just one type of antibody, which will interact with that particular epitope of the antigen. Also, some of those B cells would be set aside in the lymphatic system as memory B cells. So those are ones that have interacted with that antigen, and those are saved for future infections by the same antigen or organism. Some of these cells then enter a different zone in the lymph node called the dark zone, and that's where you get clonal expansion of those B cells. So they divide, and then there's another type of dendritic cell that then selects out these T cells that go on to induce memory B cells, as well as to create additional plasma cells which release large quantities of that antibody. So the result of all this then is you get selection and clonal expansion of a B cell that will form the correct type of antibody to react with the antigen that's on the surface of those invading organisms. So you get those will then become plasma cells, some of them, and those will release large quantities of the antibody into different fluid systems in the body. And a certain number of cells, B cells, that have that particular antibody on their surface that will react to that antigen are set aside as memory cells for future infections by that same organism. In addition to the immunoglobulins, there are many other molecules and receptors that are involved in the immune system. So up at the top, there are different immunoglobulins. There's the MHC receptors, major histocompatibility receptors on the cells. Cytokines, including interleukins, interferon, tumor necrosis factor, tumor growth factors, etc. Cytokine receptors that attach those cytokines, and then T cell receptors, and as complement system factors as well. So many different molecules are involved, a highly complex and organized system. Here's a diagram that shows you some of the rearrangements that occur in the heavy chain, in this case, in the immunoglobulins, in the B cells. So they start out in the germline having a large locus that includes at least 150 V or variable segments, and then these D and J segments for the that also wind up on the ends of these molecules, and then a constant region here. So upon signaling by antigen presenting cells to these cells, they begin to mature. So they're developmentally changing, and the changes involve recombination in this locus. So there's a recombination event choosing some of the D and J segments here. So those things that are cut out are lost. And then there's other joining between the variable region and the DJ region. There also can be selection for the constant regions. Only 3 are shown here, but there are often more than that. And so you wind up after transcription and splicing with this, and that is the only RNA and protein that will be formed, that the only type of immunoglobulin that will be formed by that B cell after that, because those recombination events are dead end. That is, most of the segments that are cut out during those recombination events are lost. They no longer are available for transcription. Similar recombination events occur in the light chains also over here, although they do not have the D region. So they have the variable regions, the J or joining regions, and the C regions here, but they do not have the D region. So they line up with the heavy chains also, lining up the same or similar regions, and then there are disulfide bridges holding those together in the final conformation. So then the constant regions here interact with the host cell, the host B cell. And these variable regions out here, both the light and the heavy chain, then interact specifically with the epitopes of the antigen. So here are the epitopes, or the surface regions of those proteins of the antigen, which is normally a protein. And you can break it down even a little further in that there are constant regions here that again interact with the host cell membrane, so they're attached to the membrane, at least in the memory B cells, not in the plasma cells where these are released from the membrane. And then there are variable regions, and way out at the end where there's that interaction with the antigen, there are hypervariable regions that interact specifically with those epitopes. These molecules join together in different ways to form different types. One of the main ones is the IgG, but IgM, which is then joining several of these immunoglobulins together, is also very common. And then some of the other ones are less common and specific to certain types of epitopes. So on the upper right, too, you can see what these molecules as a 3D model look like, where there's a constant region of the heavy chain, and then going up into these variable regions, and then the colored ones in red and brown. Those are the light chains that are tied together with disulfide bonds to the heavy chain. And these are showing just a few more details about that. So there's a constant region here of the heavy chain on both sides. These are tied together by disulfide bridges. And there are also regions here that bind to the complement region, and then the Fc receptor of the cell. So there are several regions here that interact with host proteins in those organisms, in this case in humans. Then there's this hinge region, so you get this bent section in the protein, and then a variable and hypervariable regions on the ends that are similar to those regions on the light chain, which again, and those are tied together to the heavy chains by cysteine disulfide bridges. And again, you can see this section down here that says biologic activity mediation. So those are the regions that are interacting with host cell components, including the complement system and the Fc receptors on the antibody cells themselves, the membranes. And then these ones, antigen, are out here, are involved in antigen binding, especially out in these variable regions. As I indicated, there are other receptors, and so these receptors are types of things that interact with inducers on the outside of the cell, and then it's a way of signaling those cells to become active in one way or another. So this is the interleukin type 1 family that will interact with interleukins 2, 6, and 12. And this will then send a signal inside the cell to start signal transduction and activate transcription. This one here, these interleukin 2 type, will respond to interferon or interleukin 10. These are cytokines. And those will phosphorylate certain proteins, and again start a cascade effect essentially for signal transduction that will also stimulate the cell primarily to cause cell proliferation. And then these ones, tumor necrosis factor and the immunoglobulin, family will interact with either tumor necrosis factor or interleukin 1, and these then will activate the mitogen activated protein kinases, which will then increase transcription and cell activity and cell proliferation. And then this NF-kappa B, these are nuclear factors also that will act as enhancers to increase the transcription of specific genes. So all of these will activate transcription, signal transduction, and cell proliferation. So these are all interacting with these cytokines, which are activators of those cells. So these receptors are not on all cells. These are just on cells that interact in the immune response. So here's one example of a T cell. So these are T cell receptors here. Some interact with DNA, some interact with RNA, some interact with glycoproteins, and some interact with double-stranded RNA. And they'll send signals in through a signal transduction pathway to increase inflammation by increasing cytokines and chemokines. So that's a response that then would signal other parts of the immune system and start to react to this viral infection. However, as I mentioned in the cancer, one of the cancer videos, some viruses can actually introduce viral DNA into the genome of those cells and produce a microRNA that will then become incorporated into a RISC complex, those RNA silencing complexes. And that can slow down translation of both viral mRNAs and cellular mRNAs. So in that case, it can actually slow down the inflammatory response and slow down the virus production. And in essence, in some cases, can kind of hide out from the immune system, slow down the immune system so there's not such a heavy reaction against those viruses. And this can result in a latent infection by the virus. That is, they can pretty much stay in that cell for long periods of time without the immune system reacting to them. So here are a few more details about some of the receptors. So there's an interleukin 2 receptor produced by T cells and dendritic cells. And those are then become the targets for interaction with T cells, B cells, and natural killer cells. Interleukin 4 is produced on the surfaces of T cells, natural killer T cells, eosinophils, mast cells, and that then interacts with T cells, B cells, natural killer cells, mast cells, and basophils. And then you can read through the rest of these as far as what they're produced on, what cells produce them, and what those then react with. And in many cases, you can look through here, and there are various combinations. So by releasing certain cytokines at certain times, you can have selective interactions between specific types of cells of the immune system. And here's a series of receptors that will attach interleukin 7, 2, 15, and other types of cytokines. And then they will inside the cell, there will be signal transduction, producing these JAK proteins, which will then interact with these STAT proteins, and these are transcription regulators in the nucleus, and so those will then turn on specific genes at certain times, depending on what signal is being sent to them from the cell surface receptors. And here are a few details on how that works. So you can attach the ligand out here, the interleukins, tumor necrosis factor, etc. Those act to attach to these receptors, and then the receptors on the inside will release JAK, which are kinases, or some other molecules involved in signal transduction. Those will phosphorylate the STAT proteins, and the STAT proteins then move into the nucleus and act as transcription factors to increase the transcription of specific genes, in some cases to form different receptors or interleukins or interferons, essentially to activate that cell in the immune response. So starting with a naive T cell up here, which is signaled by a dendritic cell or any antigen-presenting cell that then will stimulate that one to start differentiating into a primed T cell, which will then, depending on what the signal is that it received, will produce either interferons or interleukins or other cytokines, which will then interact with other T cells, including T helper cells type 1, type 2, type 17, and others. Those will then signal certain transcription factors, such as the STATs that I showed in the last slide. And these are transcription regulators, so these will turn on certain genes, in this case to form different interleukins, interferon, a tumor necrosis factor. And by producing these, then these will signal other cells of the immune system to act on killing viruses and killing bacteria. These Th2 cells will produce primarily different interleukins, and those will signal other parts of the immune system, including macrophages and other dendritic cells and T cells, to kill extracellular parasites. This one down here will produce other types of interleukins, primarily to increase inflammation and autoimmunity by signaling other cells in the immune system, including basophils, neutrophils, etc. And then these ones over here will produce other interleukins to activate other groups of T cells and to activate B cells also to start proliferating. Because each of the cell types has different cell surface receptors on it, embedded in its membrane, then the combination of interleukins, interferons, tumor necrosis factors, etc., these cytokines, will have different effects on different cell types. So even one of these, interleukin-6, in combination with other cytokines and other molecules in cells, can cause cell proliferation in some cases, while it can inhibit growth of other types of cells. It can have regenerative effects, for instance, in liver tissue. It can cause differentiation of certain types of blood cells. It can also cause differentiation of cytotoxic cells and other types of cells as well in the immune system. So even one cytokine can have wide-ranging effects depending on the receptors on other cells in the body. So this is the end of part one. I think you can already see that this is a very complex system, the immune system, consisting of many types of cells and many types of immunological molecules and interactions. And so I'll continue with some of these interactions in part two. So, okay, thanks very much. Talk to you later.